Infrared spectrum of 4-hydroxy-2-pentanoic acid. Structure of the organic molecule. First, examine the spectrum for major peaks. Look for characteristic absorption peaks that correspond to the functional groups present in the 4-hydroxy-2-pentanoic acid molecule. Next, identify the functional groups present in 4-hydroxy-2-pentanoic acid. Consult reference tables or databases to match observed peaks with known functional groups. Then, analyze the fingerprint region of the spectrum, typically between 1,400 to 600 cm-1. This region contains complex overlapping peaks that provide unique information about the molecule's structure. After that, evaluate the absorption peaks related to bond types. Infrared spectroscopy provides information about bond vibrations, including stretching and bending motions. Next, analyze the relative intensities of peaks. The intensity of an absorption peak can provide information about the number of bonds involved in the vibrational motion. More intense peaks indicate stronger vibrations and can help determine the abundance or concentration of specific functional groups. Then, compare the obtained spectrum with reference spectra of known compounds. Various databases and spectral libraries are available for this purpose. By matching the observed peaks and patterns with those of known compounds, you can narrow down the possible structure of the molecule. Lastly, verify with other spectroscopic techniques. This infrared spectrum shows 6 significant peaks. One of the peaks is in the fingerprint region. Firstly, there is a broad, strong peak at 3550 cm1, showing the existence of OH stretching bond, alcohol. Second, there is a broad, strong peak at 3000 cm1, showing the existence of OH stretching bond, carboxylic acid. Next, there is a strong peak at 1760 cm1, showing the existence of C=O stretching bond, carboxylic acid. There is a weak peak at 1670 cm1, showing the existence of C=C stretching bond, alkene. We also found that there is a medium peak at 1450 cm1, showing the existence of CH bending bond. In the fingerprint region, there is a strong peak at 1100 cm1, showing the existence of CO stretching bond. But this data is not reliable.
Mass spectrum of 4-hydroxypen-2-enoic acid. This is the structure of 4-hydroxypen-2-enoic acid. During the ionization process, the atom or molecule is ionized by knocking one or more electrons off to give positive ions. The atom or molecule can be ionized in either hard or soft ionization method. The ionized molecules are energetically unstable, and some break into smaller pieces called fragments. Then, the positively charged particles will be accelerated, deflected, and detected by the mass spectrometer, hence producing the mass spectrum. The base peak, which is the tallest peak in the mass spectrum, indicates the ion with the highest relative abundance. According to the bond energy table, the bond energy of the CO bond is the lowest compared to the other bonds. Therefore, it is the easiest bond to break, thus forming the most abundant ion in mass spectrum. Next, the molecular peak is the peak that represents the heaviest ion and corresponds to the molecular weight of the single charged molecule. Where the fragment peak is the peak that represents the relative abundance of the fragment ions. This is the bond energy table. Here is the possible fragments of 4-hydroxypen-2-enoic acid. Mass spectrum of 4-hydroxypen-2-enoic acid.
Nuclear magnetic resonance, NMR, for 4-hydroxy-2-pentanoic acid, presented by Cho Hui Cheng SA21086. The structure of 4-hydroxy-2-pentanoic acid was shown in slide. 4-hydroxy-2-pentanoic acid, C5H803, is an weak acid that has a molecular weight of 116.11 gram per mole. How to elucidate this organic molecule by using nuclear magnetic resonance spectroscopy, NMR. First, preparing the sample by dissolving C5H8O3 in a suitable solvent such as deuterated chloroform, CdCl3, or deuterated dimethyl sulfoxide, DMSO-D6, that does not interfere with the NMR measurement. Next, place the prepared sample in an NMR spectrometer and acquire the NMR spectrum since NMR applies a strong magnetic field and radio frequency pulses to the sample, causing the nuclei within the molecule to resonate and emit signals. Then, analyze the spectrum by examining the NMR spectrum and identify various signals corresponding to different nuclei in the molecule. By comparing these chemical shifts of functional groups, the presence of specific functional groups in the molecule can be deduced. Last but not least, determine the coupling patterns where the important structural information about the number of nearby protons, or other nuclei, and their relative positions in the molecule found by examining the splitting patterns. By observing the 4-hydroxy-2-pentanoic acid, the hydrogen ions were labeled out as HA, HB, HC, HD, HE and HF with different color. First, it can be observed that HA will influenced by the neighboring HB can causing the formation of two peaks in the spectrum. Then, HB will influenced by two neighboring HA and HC. Since HA and HC were in two different chemical environment, thus 4 peaks will generated in the spectrum for HB. For HC, it will influenced by HD and HC and giving out multiplet peaks in spectrum. HC did not influenced by HF because the distance between nuclei should within 3 sigma bond. Next, HD was influenced by neighboring HC and produced 2 peaks in the spectrum. Same as the HC that HD did not influenced by HF because the distance between nuclei should within 3 sigma bond. For HE and HF, there will be only 1 peak for each nuclei presented in spectrum because both of them are deshielded. Here is the spectrum of nuclear magnetic resonance, NMR, for 4-hydroxy-2-pentanoic acid, C5H8O3. That's all, thank you.